Hello everyone. Let me ask you something. Have you ever stood in a garden and noticed the butterfly fluttering by, a dog wagging its tail, or a fish swimming in a pond? They all look so different, yet we know they all belong to the animal kingdom. With over 1 million animal species on Earth, how do biologists make sense of such diversity? The answer is classification. Classification is the scientific method of grouping animals on the basis of shared features, just like a library arranges books into categories. This system allows us not only to identify species but also to assign newly discovered animals a proper place in the biological hierarchy. The truth is, beneath their differences, animals share certain fundamental body features, and these features form the basis of classification. Now let's explore how animals are organized at the cellular level. Even though all animals are multicellular, the way their cells are arranged varies greatly. Take a sponge, for example, its cells are loosely arranged, showing what we call the cellular level of organization. In cnidarians like Hydra, cells that perform similar functions group together to form tissues. This is the tissue level. In flatworms, or platyhelminths, tissues come together to form organs. This is the organ level. And in higher groups like annelids, mollusks, arthropods, and chordates, organs coordinate to form entire systems. This is the organ system level of organization. Notice how the complexity rises step by step, from simple to advanced, allowing animals to perform specialized life processes more efficiently. Have you ever looked at your reflection in the mirror and noticed how the left side of your body mirrors the right? That is symmetry at work. Some animals, like sponges, show no symmetry at all. We call this asymmetry. In coelentrates, ctenophores, and starfish, the body can be divided into identical halves by several planes passing through the central axis. This is radial symmetry, perfect for animals that interact with their environment from all sides. On the other hand, annelids and arthropods show bilateral symmetry. Only a single plane divides the body into left and right halves. This symmetry supports directional movement and active lifestyles. Isn't it fascinating how the body's design is linked to how the animal lives in its environment? Now, let's move into the hidden world of embryonic development. When an embryo forms, it creates layers of cells that will give rise to different tissues and organs. Animals like Hydra are diploblastic, meaning they develop from two layers, an outer ectoderm and an inner endoderm, with a jelly-like mesoglia in between. But from flatworms onward, animals are triploblastic. They have a third layer called mesoderm, sandwiched between ectoderm and endoderm. This mesoderm is critical. It gives rise to muscles, bones, blood, and many internal organs. Think about it, your muscles contracting when you run, or your heart pumping blood, all trace back to this embryonic mesoderm layer. The evolution of the mesoderm was a game changer in animal complexity. Let's now peek into the animal body cavity. Imagine your own abdominal cavity, where organs are neatly suspended. That's similar to what biologists call the colon. A colon is a body cavity lined by mesoderm, separating the body wall from the gut. Animals like annelids, mollusks, arthropods, and chordates are true coelomates. They possess a complete, mesoderm-lined cavity. In roundworms, the cavity is only partially lined with mesoderm, hence they are pseudocoelomates. Flatworms have no cavity at all and are called acoelomates. The presence or absence of colon plays a huge role in how complex the animal's body design can become. It's like having room in your house. The more space you have, the more specialized rooms you can build. Have you ever picked up an earthworm after the rains and noticed how its body looks like it's made of repeating rings? That's segmentation, also known as metamerism. In segmented animals, the body is divided both externally and internally into repeating units. Each segment may have repeated structures like nerves, blood vessels, or excretory organs. This arrangement makes movement more efficient and also provides backup. If one segment is injured, others can still function. Segmentation represents an evolutionary innovation that allowed for greater flexibility, coordination, and specialization of body parts in higher animals. And now we arrive at one of the most defining structures in animal evolution, the notochord. During embryonic development, a rod-like structure forms along the back called the notochord. This structure is mesodermal in origin and provides skeletal support in primitive animals. In chordates like us, the notochord gets replaced by the vertebral column or backbone in adulthood. Isn't it amazing? The backbone that helps you stand upright and protects your spinal cord originated from this embryonic structure. The presence or absence of notochord is so fundamental that it divides the entire animal kingdom into chordates and non-chordates. This simple rod marked a turning point in evolution, leading to the rise of complex vertebrate animals, including humans. So let's quickly bring together all the fascinating points we've covered. We began with the idea of classification, how it organizes the vast diversity of animals. We then explored the levels of organization, symmetry, and the layers of embryonic development. We saw how the presence or absence of a coilom influences complexity, how segmentation brings efficiency, and finally, how the notochord defines chordates. These features aren't just labels in a textbook. They tell the story of evolution from the simplest sponges to the most advanced vertebrates. Isn't it remarkable how a few fundamental traits can reveal the grand design of life? Remember, every time you see a worm wriggling, a fish swimming, or even feel your own backbone, you are witnessing the marvels of classification. Biology truly is the science of life in all its elegance and wonder. Until next time, when we explore more secrets of the living world.